So I thought I'd take a different approach this time around, and so I'm out here with beautiful scenery behind me, and today we'll talk about proteins. Proteins are perhaps one of the most crucial molecules in the human body for the different functions they serve. Proteins are also used in making enzymes, or should I say enzymes are proteins, and so enzymes control all the chemical reactions in the human body. As well as that, proteins are also used in chemical coordination. You might have not known that, but polypeptide hormones are proteins. As well as that, proteins, as we most commonly know, are used to repair and build tissue, cartilage, skin, and it becomes apparent that proteins are essential. But first the question arises, how are proteins made? Well, there are perhaps two answers to this question, one being a short 3-word answer, whereas the other needing a bit more information. So let's start off with the easy one first. Well, proteins are made in 2 stages called translation and transcription. And here's a long one. As previously said, protein synthesis is dictated by transcription and translation. Transcription occurs in the nucleus of a cell, wherein a chromosome, part of the DNA double helix, unwinds and unzips so that the 2 strands separate to expose the bases along the template strand. Then the RNA polymerase, an enzyme, joins up the free nucleotides with their complementary base pairs to form a complementary mRNA strand. The mRNA strand then leaves the nucleus via the nuclear pores to the cytoplasm. Now, before we proceed any further, let's first establish the differences between RNA and DNA. DNA is a double helix, whereas RNA is a single strand. DNA contains the sugar deoxyribose, whilst RNA just contains ribose. RNA contains the base uracil instead of thymine, like in DNA. Now, the way that the nucleotides line up is heavily dependent on Chargaff's rules and the base pairing rules. Now, the table on screen now shows the differences between the two, and the exact notion mentioned before can be seen, as uracil sticks out like a sore thumb, and that's in the case of thymine. Okay, so coming back now, once the mRNA molecule has left the nucleus to the cytoplasm, the DNA helix then zips up. The mRNA's code is then converted into a protein in a process called translation at the ribosomes. By this stage, the code in the mRNA consists of 3 bases, with these triplets of bases called codons. Each codon codes for a particular amino acid, and so all the codons work together to provide a code for an entire molecule of protein. Now, another molecule comes into play, that being tRNA. Each tRNA molecule has an anticodon of 3 bases at one end of its molecule, which is complementary to a particular codon on the mRNA. At the other end of the molecule is a site where a specific amino acid can attach. So to put it simply, there is a particular tRNA molecule for each and every type of amino acid. The tRNA molecule carries its amino acids to the ribosomes, where its specific anticodon pairs up with the 3 bases of the corresponding mRNA codon. Okay, so now that we've taken a look at the tRNA structure and its implication, let's take a look on how it really plays a part in translation. Firstly, the mRNA attaches itself to the ribosomes. This exposes 6 bases, or 2 codons. Then a tRNA molecule with a complementary anticodon will temporarily bond to the first codon on the mRNA molecule. A second tRNA molecule with also the complementary anticodon will then temporarily bond to the second codon on the mRNA molecule. By now, the 2 amino acids produced will join together. Meanwhile, the first tRNA molecule will leave and the third tRNA molecule will join, bonding its amino acids to the first 2. In this way, a long sequence of amino acids will join together to form a protein. Now, here's a short summary of the entire process in just 2 sentences. The order of bases in the template strand of the DNA forms the genetic code. The code is converted into the sequence of bases in the mRNA, and in the cytoplasm, the sequence of mRNA bases is used to determine the position of amino acids in a protein. So that's it, guys. I think that's a lot for today. I was going to do mutations at one point, but I've written the script for it too, but I feel as if I won't have the time for it, and it's almost about to be iftar, as you can see from the sunset behind me. Not the sun, but it's sight. So thank you guys, see you later.